Thank you for inviting me to speak today. I'm going to talk to you about our genomic-based risk assessment of avian influenza and how we use that to respond to a recent human case in the UK. So just a bit of background on avian influenza for those of you that kind of aren't familiar. Avian flu is either categorized as high or low pathogenicity, and that's defined based on its ability to cause severe disease in chickens. And it's primarily determined by a kind of cleavage motif in the HA segment and not really by much else. As everyone will know, the influenza is kind of subtyped based on HA and NA. But H5 is also classified into clades using the hemagglutinin phylogeny. And the clade 2344B is currently responsible for kind of a global panzootic. So this clade emerged in 2020 to 2021, and it was quite different to what we'd seen before in avian influenza because it wasn't really managed by normal animal health practices. So culling on farms or housing orders didn't really seem to kind of reduce the burden. And we also saw a kind of expanded host range. We were seeing lots more shorebirds, migratory birds, and this contributed to a global spread. The clade also caused quite a lot of mammalian infections, and some of those were quite big outbreaks. So you might be familiar with some of the fur farm outbreaks in Europe, or the pinniped outbreaks in South America, and most recently the cattle dairy farm outbreaks in the US. Alongside that, there were quite a lot of human infections. So in the tree on this slide, all the human infections are kind of in red, which hopefully you can still see, but they're kind of across the tree, and there were quite a lot of them within this kind of 2344B clade for people who were kind of working on farms or were in environments that were contaminated. So how do we actually assess the risk to a human from a sequence that came from a bird? It's unfortunately not as simple as just looking for a cleavage site in HA, but we're primarily looking for 4 viral characteristics. The first one is polymerase adaptation, and that is kind of mutations in any of the 3 segments that are involved in the polymerase complex, and the mutations that we see in these segments will increase replication in mammalian cells. We also look for resistance to host restriction factors, and these are kind of less likely to immediately appear in a mammalian host and are sort of more genotype to genotype specific, but are based on caused by mutations in the NP segment, and will basically just mean that your kind of innate immune response is less effective if you do come into contact with this virus. And the last 2 are the most important, but also the most complex, and they are based on the HA segment and the kind of host specificity of that. So we look for changes in the kind of receptor binding specificity across HA, and that is highly host-specific, but also tissue-specific. So you will see different tissues that are infected in different people based on this receptor specificity. So in birds, you see what we call alpha-2,3 linked sialic acid receptors, but in humans, we have what we call alpha-2,6 linked receptors. And for effective infection and host cell entry, but also between host transmission, you need to have these kind of receptor-specific changes in HA. The other thing that HA needs to be able to do is persist in different environments. So the core temperature of a duck is lower than the core temperature of a human. So the HA kind of mutates to survive at different temperatures and kind of persist in different parts of those hosts. In birds, it tends to be more of a gastric infection, and so their transmission is kind of fecal-oral route, but for humans, it's obviously a respiratory infection, and we see airborne transmission. But you don't really get that airborne transmission for avian influenza because it doesn't have these HA changes. So into the meat of the actual kind of assessment that we did within the UK. So our animal health colleagues share sequence data with us routinely for anything that they generate from their own surveillance. And we then assess that based on those kind of 4 principles that I just talked about to evaluate the current risk to human health. So looking at the data that they provided us, we were able to say that in January 2025, DI.2 was the predominant genotype that we were observing in UK birds. From the publicly available global data, we were also able to say that this genotype had been predominant across Europe the previous avian influenza season. And that meant that we could kind of assess how many mammalian outbreaks we'd seen, how many human infections we'd seen, how severe all of that seemed to be. We could show that there were no core mutations present in this genotype that would affect any of those 4 characteristics that I was just talking about, and the HA was consistent with avian receptors. We had early in January, we'd had a cluster of sequences from an infected premises in the UK that did have a polymerase mutation that would be associated with mammalian adaptation. But that premises was managed by our animal health colleagues and was considered to be kind of a closed outbreak. We weren't concerned that mutation was spreading across the UK birds. So overall, we assessed the season as low risk. And that meant that we were only really managing high-risk exposures with antivirals and close contact tracing. So somebody that was exposed to infected animals with no PPE or a breach in PPE, or was exposed to a confirmed human case. And if you can manage a situation in that way, it obviously really reduces the burden on your public health teams because you've got fewer people to follow up, you've got fewer antivirals to try and kind of spread everywhere, and you've got fewer people that have to go into a high-consequence infectious disease unit in a hospital, which have very limited beds. So obviously we were notified about our UK case on a Friday afternoon, because why would it be any other time? But we were able to produce sequence data, not just from the human case, but also from the poultry on the farm within 72 hours of the human sample collection. And this was really valuable for us because we were able to show that the sequence from the human was exactly the same as what was seen in the birds. We weren't seeing any adaptations that were host-specific, and it was consistent with that DI.2 genotype that we'd previously assessed. So that meant that we could just manage this single exposed person, we managed their close contacts, but we didn't have to do any kind of immediate vaccination, any extended contact tracing, and once that person kind of recovered and had isolated, we were really only managing people on the farm that were exposed to the birds. We weren't concerned that there were kind of onwards transmission chains that we weren't going to be able to control. So you can see in the tree as well, the sequences that are labeled, they're the sequences from the chicken and the human in this situation. And they're kind of different from the rest of the UK cases, which are in purple, but still kind of clustered roughly with them. And that's really just because the surveillance in the birds is quite sporadic. We don't have a lot of surveillance across wild birds, because funnily enough, they don't submit to sampling just walking into a public health lab. And we only really see sequences from infected premises, and the kind of aim is that we would get at least one sequence from each premises, but as many as possible. But it does mean that we have to kind of infer things, and there can be quite a lot of mutations between what you've kind of assessed and analysed and what you actually see in your sort of human case. But luckily in this case, there weren't too many mutations for us to look at, and we could do that rapidly. So we had sequencing within 72 hours, and then within kind of the next day, we had all of our reports shared, and we were happy that the public health response had been proportionate, and we didn't need to upgrade any of those response efforts. So just to kind of finish with some sort of summary points, the kind of proactive assessment of this sequence data from the animals was really helpful in our rapid response to a human case. We already knew a lot about what was circulating and what that would mean in terms of our response. And that really depended on us identifying these kind of genomic features that would inform the risk to human health, and a lot of that comes from our academic collaborators. But collaboration between our animal and human health agencies is obviously super important in this. If they hadn't sent us any data, we wouldn't have been able to do those risk assessments. And similarly, kind of the global contribution to sharing sequence data, we can't tell anything about what's going on in the animals if no one shares anything with us. But the surveillance of those animals is really important. So that a few times kind of throughout the conference, we've talked about funding and prioritization of that. And I think it's really important that we don't just focus on sequencing the pathogens from the humans, but we need to think about this in a One Health way and where those infections are actually going to come from. Because if you understand that reservoir, then you understand the risk to people much better. And also just because metadata is everything, this is, I guess, just a plea for like sufficient and consistent metadata, because if you don't know what host your sequences come from, what country it was detected in, when it was collected, all of this is entirely pointless. So yeah, please include your metadata. But that's all from me. Thanks.